Welcome to the deep dive. We're here to unpack some really fascinating sources and pull out the most surprising insights for you. Today, we're tackling something pretty fundamental: memory, how we think about it. The standard picture is, well, it's all in the brain, right? Kind of personal, internal archive tucked away. But what if that's incomplete? What if memory isn't just inside our heads? What if it's like networked, multi-scale, stretching across organisms, maybe even ecosystems?
It's a pretty bold idea, isn't it? Because, yeah, the focus has always been so grain-centric, so individualistic. But we're looking at a new framework here, something we're calling symbiotic topological memory.
Hmm.
And the core idea isn't about, storage capacity or counting neurons. It's about connectivity. It's the relationships, the patterns between things, neurons, organisms, fungi, whatever it might be.
Mm.
It really challenges our, well, human-centered view. Memory is something relational, distributed.
Exactly. And our mission today is to unpack three, well, they seem totally unrelated at first glance, peer-reviewed sources. We'll start with a neuroimaging study looking at how childhood trauma impacts the brain. Then we pivot into computational neuroscience, a paper looking at how the hippocampus maps space topologically, not just geometrically, which is quite a shift. And finally, we're going underground, really, into the world of plant-fungal relationships, cognition even. And we want to show you how network topology, that's the math of connection and shape, ignoring size, how that lens reveals these hidden links that could change how we see memory, and maybe even how we approach healing from trauma.
It's quite a journey we've mapped out. From the human brain, its vulnerability,
Mm-hmm.
through these abstract mathematical ideas about memory structure, and then deep into this fungal world. It really challenges assumptions about where intelligence resides, and the surprising endpoint, maybe a new way to think about therapy, grounded in this very interconnectedness.
Okay, let's start then. Let's look inward first at the human brain and how something like childhood trauma can literally reshape our memory systems. Pillar one, trauma-induced limbic remodeling in humans. Right, so pillar one, this is a very recent study, Liu and colleagues, 2024, in BMC psychiatry. They're looking for structural differences in the limbic system related to childhood trauma.
Yeah, it's about how those early experiences, leave a physical mark, a structural imprint. So Lou's team took 48 healthy adults. Half had a documented history of childhood trauma, the other half didn't, a control group. And they used the really precise MRI technique, volumetric segmentation, to measure the size of different bits of the brain, focusing on the hippocampus and amygdala, key areas for memory, emotion, stress. Usual suspects, really.
And what did they actually find? Were there differences?
There were, yeah, quite striking ones. The group with childhood trauma, they showed significantly smaller volumes. But not just like general shrinkage. It was specific subregions within the limbic system.
Okay.
So in the hippocampus, you had parts like this subiculum head body, CA1 head, the molecular layer head, the GC-MLDG body, that's the granule cell layer, the CA4 body, the fimbria, even the area connecting the hippocampus and amygdala. And several amygdala nuclei were smaller too. It's quite granular detail.
Wow, that's specific. And did they connect those changes to the type of trauma?
They did try to do that, yeah, which is interesting. They found, for instance, that smaller volumes in the CA4 body and that GC-MLDG body seem to correlate specifically with sexual abuse history. And changes in the right basal forebrain, another area involved in memory, were linked more to emotional neglect. So it suggests, maybe different types of trauma leave slightly different signatures on the brain's architecture, potentially affecting different memory or emotional functions.
That sounds really significant, but you mentioned detail. Were there limitations? Caveats?
Absolutely. And this is crucial. The authors themselves point this out very clearly. Those specific correlations, they actually lost statistical significance after correcting for multiple comparisons. Yeah, it's a common issue. When you test lots and lots of correlations in a small group, some will pop up just by chance. So while the findings are definitely suggestive, maybe pointing future research in that direction, we have to be cautious because the sample size was relatively small. The stats just didn't hold up under stricter correction.
Okay, so intriguing, but needs more work. What about the focus on volume itself? Is smaller always worse?
That's another great point. The paper focuses on volume, and there's often this implicit idea that bigger is better, healthier. But, focusing only on size might miss the bigger picture. The topological organization, how things are connected. Think about it like, well, a city might shrink physically, but if it reorganizes its transport network really efficiently, it could still function perfectly well, maybe even better in some ways. So trauma might not just cause loss, it might trigger a reorganization, shifting the brain into a different kind of connectivity state, perhaps one adapted for survival in adversity. It's not just shrinkage, it's potentially adaptive restructuring.
So it's less about the amount of brain stuff and more about how it's wired up, how the connections change under stress. Like, you need the bricks, sure, but the structure is what makes it a house. Did Lou and colleagues mention other limitations?
They did. They were upfront about relying on self-reported trauma, which always has subjectivity issues. It was also cross-sectional, just one snapshot in time. We don't see how these changes developed. And critically for our discussion today, they didn't look at functional connectivity, how these regions were actually talking to each other. That would tell us more about the network dynamics.
Okay, so despite the limitations, it gives us a starting point, right? It shows experience, especially trauma, shapes the brain physically. It pushes us to think beyond just volume, maybe towards, well, towards topology.
Exactly. It sets the stage perfectly for thinking about what these structural changes actually mean for the network's organization and function. Pillar 2, persistent homology and topological memory in the hippocampus.
Okay, now for a bit of a gear shift. We're moving from the biological brain scans into computational neuroscience, and some pretty advanced math, actually. Our second source is this really influential paper by Dabaghian and colleagues from 2012 in PLOS Computational Biology. And it fundamentally challenges how we think the hippocampus, our memory powerhouse, maps space.
Right. The traditional view, the sort of intuitive one, is that the hippocampus works like a geometric map builder, like an internal GPS. It's plotting coordinates, distances, angles. That's how it represents space.
Seems logical enough.
It does, but Dabaghian and colleagues argue. Maybe that's not how brains actually work. Neurons don't output GPS coordinates, do they? They output spikes, complex patterns of electrical activity. So downstream brain areas reading this hippocampal output, they aren't getting precise locations. They have to decode the pattern. They need to figure out connectivity, adjacency, which cells fire together, which fire in sequence. So the argument shifts. Memory isn't about the absolute where, it's about what's connected to what, the relationships.
Wow, okay. That is a shift. So memory as relationships, connections, not exact locations or sizes. That sounds like topology.
Precisely. That's the core idea. A topological representation. Think about the classic topology example.
Yeah.
A coffee cup and a donut. Topologically, they're the same. Why? Because they both have one hole. You can squish and stretch the clay of the cup into the donut shape without tearing it. The exact geometry doesn't matter. The fundamental structure, the single hole does.
Right. The connectivity, the whole persists.
Exactly. And this paper suggests hippocampal memory work like that. It's about the persistent patterns of connection. The who fires with whom, not the exact metric distances.
So how did they actually test this idea? It sounds pretty abstract.
They used a mathematical tool called persistent homology. It's designed for exactly this kind of problem. Imagine you have data points like neuron firing times. Persistent homology looks for robust shapes and connections within that data cloud. It sort of zooms in and out mathematically, looking for features like loops or holes that persist across different scales of analysis. Things that aren't just random noise. They used it to calculate things called Betty numbers. These numbers quantify the holes in the network structure. Betty 0 is connected components, Betty 1 is loops, Betty 2 is cavities or voids, and so on. It gives you a topological fingerprint of the network.
And they applied this to simulated rat brains.
Basically, yes.
Oh.
They built computational models of hippocampal place cells firing as a simulated rat explored an environment. They could change parameters in the model firing rates, the size of the place fields where a neuron fires, the number of neurons involved. Then they looked at the patterns of co-firing when multiple place cells fired together. And they applied persistent homology to those co-firing patterns. And crucially, they found that these patterns contained robust topological information about the environment, enough to build a map and fast enough to be biologically plausible.
Okay, so the math supports the idea that topology could work for spatial mapping. What else came out of it?
This is maybe the most intriguing part. They found what they called a learning region. Think of it as a sweet spot in the parameter space, the combination of firing rates, place field sizes, neuron numbers. Within this region, the topological map forms reliably. It's stable.
Yep.
But if you push the parameters outside this region, the map formation fails. It breaks down.
Ooh.
Yeah. It implies memory formation isn't guaranteed. It depends on the system operating within certain bounds. And those bounds, those parameters, could potentially be shifted by things like stress, disease, or even just changes in the environment.
Okay, now I see the connection back to the trauma study. If trauma messes with brain function, neurochemistry, maybe it pushes the hippocampus out of that optimal learning region.
That's exactly the speculation. It could explain memory problems after trauma issues with spatial context, narrative coherence, even if the hippocampus hasn't shrunk dramatically. The dynamics, the topology are off.
And interventions then might aim to nudge the system back into that region.
Potentially, yes. Restore the conditions needed for stable topological map formation. Of course, we need to remember the limitations here, too. This is simulation-based. Real brains are way messier. And persistent homology captures certain topological features, maybe not the whole story of network dynamics.
True.
But it gives us a powerful mathematical language, a way to talk about memory networks where connectivity, the relationships are paramount. Pillar 3, mycorrhizal networks and extended plant cognition.
Right, we've gone from trauma's physical impact to the abstract math of memory maps now for something completely different. Or is it? Prepare yourself, because we're heading underground, into the world of fungi and plants. Our third source is a review by Parise and colleagues from Symbiosis, just published or forthcoming in 2025. And the idea is pretty wild, that mycorrhizal fungi, these fungi that partner with plant roots, might actually extend plant cognition.
Yeah, definitely pushes boundaries. We usually think of mycorrhiza in terms of nutrient exchange, right? Fungi are great at scavenging phosphorus, nitrogen, water from the soil. They act like root extensions. In return, the plant gives them sugars from photosynthesis. It's a classic symbiosis, a trade.
The Wood Wide Web idea, basically. Trading resources.
Exactly. But this review goes way beyond that. Parise and team are suggesting these fungi aren't just passive pipes for nutrients. They're proposing they might be active informational partners, literally expanding what we might consider the cognitive abilities of the plant.
Okay, you have my attention. How? How would fungi extend plant cognition?
The review outlines 4 key ways this might happen. First, foraging complementarity. It's more sophisticated than just general nutrient sharing. The fungus, with its incredibly fine hyphae, explores soil the roots can't reach. It can specifically target and bring back resources the plant needs right now, based on the plant's signals. It's a cooperative, targeted foraging.
Okay, that's one. What else?
Second, dynamic recruitment and abandonment. Plants aren't stuck with the fungi they first meet. They seem to actively choose fungal partners, strengthening ties with beneficial ones and cutting off those that aren't pulling their weight or are even harmful. This implies the plant is constantly evaluating and making decisions about its network partners.
Evaluation and decision-making. Sounds cognitive. What's number 3?
This one is maybe the most mind-bending. Expanded perception of the soil environment. The fungal network acts like a huge underground information network. It can relay signals to the plant about distant resources, maybe water patches far away, or about threats, like pathogens moving through the soil, or even signals from other plants connected to the same network, maybe stress signals or resource availability.
So the plant's awareness extends way beyond its own roots, through the fungus.
That's the idea. Its sensory world gets massively expanded by the network.
Okay, and the fourth point?
Fourth is shaping fungal communities. Plants aren't just passive recipients. They release chemicals, change soil conditions, and through these actions they can influence which fungi thrive nearby, actively shaping the network composition to suit their own long-term needs, like actively curating their symbiotic community. So, taken together, these 4 processes suggest the fungal network isn't just plumbing, it's acting like a memory store. It holds information about past resource finds, remembers danger zones, relays information across space and time.
Memory in a fungus network. That's pretty radical. And didn't the review mention network structure similarities to brains?
It did. They point out that these mycorrhizal networks often show small world and scale-free properties.
Which means?
Small world means it's easy to get from any point in the network to any other point, quickly short path lengths. Efficient information flow. Scale-free means some nodes may be connection points between fungi or between fungi and plants are highly connected hubs. This makes the network resilient. You can lose random nodes, but the hubs keep it connected.
And that's like neural networks.
Exactly like the structures we see in brains. It suggests a deep structural parallel between how information might be processed in a brain and in this underground fungal network.
Wow.
Now, we absolutely have to add the caveat here. This review is speculative. It's building a strong case based on analogy and existing evidence, but it's a hypothesis. The authors themselves are clear about that. They propose experiments to test these ideas rigorously. Direct proof of cognition in the system is still needed.
Understood. Speculative, but incredibly provocative. It forces you to ask, well, what is cognition? What is memory? If it can happen outside a brain in a distributed symbiotic way like this, what else are we missing?
Precisely. It challenges our assumptions and opens up entirely new ways of thinking about information and life.
Okay, so we've got trauma-changing brain volumes, maybe topology, the hippocampus using topology for memory maps, and fungi potentially extending plant memory via networks. Three very different worlds. Now let's smash them together. Collision mode. What happens when we look at them all through this lens of symbiotic topological memory?
Well, the first connection that just leaps out is reinterpreting those trauma findings from Liu et al. Through the topological lens of Dabaghian et al. Instead of just saying limbic volumes are smaller, we can hypothesize that trauma fundamentally alters the connectivity patterns, the topology. Maybe that stress pushes the hippocampal network outside its optimal learning region. So its ability to form stable topological maps is impaired, even if the volume loss isn't catastrophic. The volume change is a symptom, perhaps, of a deeper topological disruption.
Right. And those specific areas Lou found affected the GC-MLDG body CA4. They're crucial for things like pattern separation in the hippocampus. That's telling similar memories apart. So reduced volume, there isn't just less brain. It implies changes in the actual network wiring, fewer synapses, maybe altered dendritic branching, different glial cell interactions, all things that change the network's topology, its shape and connectivity, which would directly impact its ability to form distinct topological memories.
Exactly. And this suggests something really practical. Maybe topological data analysis, TDA, could give us much better biomarkers for trauma than just volume measurements. Imagine applying persistent homology not just to structure, but to functional brain data EEG or fMRI connectivity patterns. You might find specific topological signatures of trauma, maybe fewer stable loops like a lower Betty 1 number, or loops that collapse more easily, representing fragmented or unstable memory circuits.
Like seeing not just a smaller house, but seeing that the internal wiring is faulty or key connections are missing.
Precisely. A much richer, more functional picture than just size.
Okay, so trauma affects brain topology. Now, can we stretch this topology idea further to the fungi?
I think we absolutely can. The Dabaghian paper provides the language. Mycorrhizal networks, as Parise and et al. Describe, also have complex topologies. They're nested, modular, scale-free. We could apply the same mathematical tools, like persistent homology, to analyze the structure of these fungal networks. We could map out the persistent loops and cavities in the hyphal structure. What do they represent? Maybe robust pathways for nutrient flow, or protected channels for signaling molecules, or information storage hubs.
And could we then ask the same question Dabaghian asked about the hippocampus? Is there a fungal learning region, an optimal range of parameters, branching density, soil moisture, nutrient levels, where the plant fungus network processes information most effectively, where it forms the most robust memories of its environment?
It's a fascinating possibility. And what happens if environmental stress, drought, pollution pushes that symbiotic system outside its optimal topological state? Does its ability to adapt, to remember past conditions, degrade?
And thinking back to Parise's four mechanisms, foraging, partner choice, expanded perception, community shaping, could we see those as topological operations?
That's a great way to frame it. Each of those involves adding or removing connections, edges in the network, strengthening some pathways, weakening others. It looks remarkably like synaptic plasticity in the brain, changing connection strengths to learn and remember. But here, it's hyphoplasticity, chemical signaling plasticity, network architecture changes in the soil, optimizing information and resource flow. It's adaptive network reconfiguration, memory through structural change.
This leads to that memory as symbiosis idea, blurring the lines, because childhood trauma and mycorrhizal symbiosis, they seem worlds apart. One is internal, often painful. The other is external, ecological, cooperative. But both involve the environment leaving a lasting imprint on an organism's network structure, right?
Exactly. Trauma disrupts internal neural networks. Mycorrhizal symbiosis enhances and extends networks externally. Both fundamentally challenge the idea that memory is purely an internal, individual thing. We could even frame childhood trauma as a disruption of an ecological memory scaffold. Think about it. A safe, predictable environment provides external stability and external memory that helps the internal memory systems develop resilience. Trauma removes that scaffold.
So the external environment helps build the internal memory structure. And plants, they seem to almost outsource cognition to the external fungal network.
It looks that way. Outsourcing perception, information gathering, maybe even some decision-making. In both cases, human and plant, the boundary between internal memory and external environment gets really fuzzy. It echoes those extended mind ideas. Cognition isn't just in the head, it extends into our tools, our bodies, our surroundings. Here, the surroundings are literally alive and networked.
But there's a tension, isn't there? Trauma feels so intensely individual, a personal burden, while the fungal network sounds like collective memory, shared information, community benefit.
That's a key tension. But maybe human memory isn't purely individual either. Think about our reliance on social networks, cultural knowledge, family stories, even our gut microbiome influencing our mood and memory. These are all external scaffolds for our internal memory.
So trauma might not just damage the brain, but also cut people off from these collective memory networks, leading to isolation.
It's a strong possibility. And maybe resilience, then, in both humans and ecosystems, comes from that topological feature, redundancy, having multiple pathways, strong loops, dense connections, whether it's social support networks for a person or diverse mycorrhizal links in a forest. These robust topological features allow the system, individual or collective, to withstand shocks and maintain function. Redundancy is resilience. Hashtag critical counterpoint in visionary mode. 1. Critical counterpoint, moving from speculation to empirical evidence.
Okay, we've built this really compelling, interconnected picture, but it rests on these three pillars, each with its own limitations. We need to be critical. How do we move this beyond intriguing parallels and towards solid, testable science?
Good point. Let's start with Luc et al. On trauma. We already flagged the sample size and the multiple comparison issue. That weakens the specific claims about which regions link to which trauma types right now.
So what's the next step for that line of research?
Definitely larger studies, and longitudinal ones following people over time, but more importantly, move beyond just volume. Apply TDA, persistent homology, to functional connectivity data from trauma survivors. Use fMRI or EEG to map their brain activity patterns into these high-dimensional connectivity spaces. Then look for those topological signatures, the holes, the loops, their stability. That could reveal subtle network reorganizations linked to trauma symptoms, providing a much richer picture than just saying this bit is smaller.
Okay, makes sense. What about the Dabaghian paper, the topological hippocampus model? It's mostly simulation, right?
It is. Beautiful theory, elegant math, but largely theoretical. The key next step is validation with real biological data. We need to record from actual hippocampi, probably in rodents initially, while they're learning spatial tasks, and apply persistent homology to those real neuronal firing patterns.
And compare that to the model's predictions.
Exactly. Does the topology of the real firing patterns match the theory? Does it predict the animal's behavior? You could even do pre-post studies. Record activity, introduce a stressor, record again. Does the stressor disrupt the topological structure of the map, as predicted by the learning region idea? Does that disruption correlate with memory impairment? And ideally, link it to structure too. Combine the topological analysis with detailed imaging of neuron shapes, dendrites, bridge the functional topology to the physical structure.
Right, linking levels. Now, the plant fungi cognition paper, Parise et al., highly provocative, but you stressed it's speculative.
Very much so. The analogies are powerful, but we need direct evidence that information transfer through fungi is mediating complex cognitive-like behaviors in plants beyond just basic signaling or resource exchange.
So how do you test that experimentally?
You need clever experimental designs. Combine network analysis with behavioral tests. For example, set up plants with different fungal partners, some known to form complex networks, some simpler ones. Control the nutrients very carefully, then give the plants foraging choices or expose them to localized threats. Do the plants with more complex fungal networks make smarter decisions? Do they respond more effectively? You need to map the fungal network topology using imaging and TDA and correlate changes in that topology over time with the plant's behavior.
And maybe manipulate the network.
Yes, use genetics or chemicals to block specific signaling pathways in the fungi. If you block a pathway thought to carry information, does the plant's supposedly cognitive behavior disappear? Does the network topology change predictably? That's how you move from correlation and analogy towards establishing actual causal links. It's hard work, but necessary. 2. Visionary mode. Cranking the weirdness, bold speculation, grounded in science.
Okay, critical checks done. Now let's embrace the weirdness again, but grounded weirdness. If this symbiotic topological memory framework holds up, where could it lead? What are the really far-out implications?
Well, one thing is these fractal echoes across scales. It's fascinating that both neurons with their dendritic trees and fungi with their hyphal networks show fractal patterns self-similar branching at different magnifications. Trauma might disrupt dendritic fractality in the brain. Fungi adjust their fractal branching based on resources. And guess what? Persistent homology is actually pretty good at detecting fractal-like structures via how topological features persist across scales.
So Memory networks, brain or fungal, might follow these universal, scale-free, fractal principles.
It seems plausible. And maybe resilience, the ability to handle damage or noise, depends on maintaining that fractal integrity. Disrupt the fractal pattern, and maybe you disrupt information processing capacity.
And what about extending that learning region idea?
Right, beyond the hippocampus. Could we define an optimal learning region for a whole plant fungus network? A specific range of parameters, fungal density, soil chemistry, water availability, where the symbiotic system is maximally effective at processing information, adapting, remembering.
And stresses like pollution or drought could push the whole ecosystem outside that optimal region, reducing its overall intelligence or adaptability.
Exactly. Understanding that could be huge for ecology. Imagine managing ecosystems not just for biodiversity, but for their cognitive resilience, their topological health, keeping them within their learning region to face climate change.
That connects to something else, these trauma ecology feedback loops.
Yes, it's becoming clear that human trauma and ecological damage are often linked. Climate disasters cause trauma. Deforestation causes displacement and grief. And traumatized populations might, understandably, engage in behaviors short-term thinking, resource over-extraction, apathy that worsen the environmental damage. It's a vicious cycle.
But the reverse could also be true. Healthy ecosystems support mental health.
Plausibly, yes. Stable environments, biodiverse soil microbiomes influencing our own gut-brain axis, places for connection and healing. This framework suggests a direct feedback. Internal trauma disrupts our networks, leading to behaviors that damage external networks. Degraded external networks then offer less support, less resilience, less ecological memory for us to draw on.
So healing requires working on both levels, personal and planetary.
It strongly implies that, yes, they're intertwined.
This is getting pretty profound. Any final weird thoughts? AI, philosophy.
Well, it does resonate with some philosophical ideas, doesn't it? Panpsychism, process thought. The idea that mind or memory isn't exclusive to brains, but maybe a more fundamental property of complex network systems. Finding memory-like processes in fungi, seeing topology is fundamental, it lends a certain scientific plausibility, or at least resonance, to those older intuitions about a mind and nature.
Not saying fungi are conscious, but that the processes we associate with mind are broader than we thought.
Exactly. And keeping that humility is important. It could even inspire AI. Instead of building isolated AI brains, what about AI that learns symbiotically, that interfaces deeply with environmental networks, drawing data and adapting topologically like these natural systems seem to do? AI that's embodied and ecologically embedded.
Okay, wow. From fractal fungi to mindful AI. Let's pull this back down to earth. How can this synthesis actually help someone, say, recovering from trauma? You mentioned a new therapeutic approach.
Yes. Based on this synthesis, we're proposing something we call ecological topological therapy, or ETT. This isn't just talk. It's a concrete, practical intervention designed from these principles.
Ecological topological therapy, what's the core idea?
The core premise is that healing from trauma, deep healing, requires more than just internal work. It needs a conscious reconnection between the individual's internal neural networks and the external living ecological networks around them. It's about bridging the disconnect trauma often creates, using the environment as an active partner in rebuilding internal topological resilience.
Okay, sounds fascinating. How would it actually work? What are the components?
We envision three tightly integrated components. First, mycorrhizal inoculation and green spaces. We'd create specific therapeutic gardens, topological gardens. These wouldn't just be pretty, they'd be designed and inoculated with diverse mycorrhizal fungi known to create complex, resilient networks.
So, gardening therapy, but supercharged with fungi?
Kind of, but much more focused. Patients would actively participate, planting, tending, observing these plant-fungus partnerships, knowing what's happening underground. The garden design would aim for maximal network complexity, diverse plants, dense fungal connections, and crucially, we'd embed sensors in the soil.
Sensors? For what?
To monitor the external network in real time. Soil moisture, nutrient flows, maybe even electrical activity or growth patterns in the hyphal network. We quantify the health and complexity of the garden network itself.
Okay, so you have the living garden network. What's component two?
Topological monitoring. This is where the tech comes in. We collect data simultaneously from the soil sensors in the garden and from the participant's brain, using maybe portable EEG or fMRI in research phases to capture their neural functional connectivity patterns.
And then you analyze both sets of data.
Exactly. Using persistent homology algorithms.
Yeah.
In real time. The output would provide feedback on the topological richness, the complexity, the number of stable loops, the resilience of both the person's internal brain networks and the external garden network they're interacting with.
Wow. Real-time feedback on brain topology and garden topology. How does that translate into therapy? That's component 3.
Yes. Biofeedback and narrative integration. The participant gets this biofeedback, seeing how their internal state correlates with the garden's network dynamics. Maybe they see their brain's network complexity increase during mindful moments in the garden, or they see how stress might temporarily disrupt both internal and external patterns. It makes the connection tangible, embodied.
I see.
And therapy sessions would weave this together with narrative work, helping people externalize traumatic memories, maybe mapping them onto the garden, like describing a feeling of fragmentation corresponds to seeing a less connected part of the fungal network, and then actively working on nurturing that part of the garden becomes a metaphor, maybe more than a metaphor, for internal reintegration. It's about rebuilding coherent narratives of self in relation to these living networks, healing through reconnection, documented topologically.
That's incredibly ambitious. How would you even test if it works?
You'd need a proper palette study. Maybe 30 adults with childhood trauma history, randomly assign half to ETT, half to a standard therapy control group, run it for, say, 12 weeks.
And measure.
Standard things like PTSD symptoms, sure. But crucially, also measure changes in hippocampal connectivity using those TDA metrics we talked about, Betty numbers, loop stability, and track the measured topological complexity of the fungal networks in the ETT gardens. See if internal and external network health improve together, and if that correlates with symptom reduction. Obviously, strict ethical protocols are paramount trauma-informed care, screening for any risks from fungal exposure, data privacy. It's complex, but potentially transformative. #, conclusion outro.
So quite a journey today. From brain scans of trauma survivors, through the mathematical beauty of topological memory maps, to the hidden cognitive world of plant fungal networks. And bringing it all together, we arrive at this idea: symbiotic topological memory. It really asks us to reconsider memory not just as files in our head, but as this dynamic network property emerging across scales, across life itself.
And the key, the thread connecting it all, is topology. That mathematical language of connection, of relationships. It gives us a way to see the fundamental similarities between how a brain might process trauma and how a forest ecosystem might store information or build resilience. It's about the patterns of connection.
And the big takeaway, maybe the most hopeful one, is this idea that healing, learning, adapting, it's all about reconnection. Reconnecting our internal networks, often fragmented by trauma or stress, with the supportive, complex, living networks outside ourselves, closing that gap between self and world.
And ecological topological therapy, ETT, is our attempt to make that concrete. To build a therapy that actively uses nature's networks, monitored through this topological lens, as a co-therapist in the healing process.
It's a profound shift, acknowledging this symbiotic, topological view of memory. It could change more than just science. It might foster new ways of thinking about community, about our place in the world, maybe even a deeper spirituality grounded in interconnectedness. Definitely a new sense of stewardship. So the final thought for you to ponder: what does it really mean for you, for your sense of self? Your memory isn't just yours alone, but is woven into the very fabric of the living world around you.